Hi, it's Dr. Phoenix. Today we're going to look at the protein synthesis drag and drop activity. I've created an Excel spreadsheet that's on My Big Campus and on the website where you can practice protein synthesis in a drag and drop activity. In this video I'm going to show you how to go about doing that. Remember that protein synthesis is a two-step process. Transcription is where a gene which codes for a particular protein is transcribed into a messenger RNA molecule, and the second stage is translation, where that messenger RNA molecule is now translated into a protein. So let's look at the first step, transcription. Remember, transcription takes place in the nucleus. In the nucleus are DNA molecules, which are very, very large molecules. They carry many genes, and each gene codes for a particular protein. These DNA molecules are so large that they can't get out of the nucleus. And we know that protein synthesis takes place out in the cytoplasm where the ribosomes are. So since DNA can't get out of the nucleus, there needs to be some method of copying the message or the blueprint of a gene that can be taken out into the cytoplasm where protein synthesis takes place. All right, that method is called transcription. And so on this page we're going to practice transcription. This oval represents the nucleus. This string of nucleotides is a strand of DNA. It is a section of the hemoglobin gene that has been unzipped during transcription. The DNA molecule is partially unzipped by helicase, not completely like in replication. It's only unzipped enough to expose that gene. Then along comes RNA polymerase. RNA polymerase will read the nucleotide base sequence on the gene and will attach free RNA nucleotides to their complementary bases to create a strand of messenger RNA. So here we have RNA polymerase. We have a couple of docking stations here for these RNA nucleotides to attach. So the first one will come in, and that will be a G that will match up with the C on the DNA molecule. And then remember, RNA has no T's. Instead of T's, RNA has U, or uracil. And so A in DNA will match up with a U in RNA. And so this RNA polymerase will travel on down the gene, matching up RNA nucleotides to the DNA nucleotides until the entire gene is transcribed and we've created a messenger RNA molecule. Once that messenger RNA molecule has been created, it's going to leave the nucleus and go out into the cytoplasm where it can be translated into a protein. So here we are out in the cytoplasm. Here comes our messenger RNA molecule that was just created in the nucleus during transcription. The messenger RNA molecule is going to find a ribosome and it's going to attach to it. There are two docking stations on the ribosome. These docking stations are to allow a transfer RNA molecule to dock and line up with what are called codons on messenger RNA. Codons are 3 base sequences on messenger RNA that code for a particular amino acid. So there's going to be a transfer RNA molecule that will have a complementary base sequence to GUG, and it will be carrying a particular amino acid that GUG codes for. So the complementary base sequence to GUG would be CAC. So CAC happens to be right here, And so CAC will be bringing valine down into the ribosome where the proteins are going to be built. Remember, proteins are built from amino acids. The next docking station is going to be reading the next codon, CAU, which will match up with GUA. GUA happens to be right here, and so GUA will come and sit down. When these two amino acids are next to each other, they will form a peptide bond. All right, the first transfer RNA molecule will leave. All right, the messenger RNA will slide on down the ribosome. All right, the second docking station is available to receive another tRNA molecule. Again, the appropriate tRNA will bring the right amino acid to its location. All right, there'll be another peptide bond that will form right here. The first tRNA will leave, and this process will continue, reading codons, tRNA matching to its complementary bases, codon and anticodon matching up. We're going to lose a transfer RNA, again creating peptide bonds between the amino acids. The ribosome will slide down. The next tRNA will come. Peptide bond will form. tRNA leaves. Ribosome slides. All right, here comes the final tRNA. Peptide bond forms. Amino acid leaves. Ribosome slides again. There goes our last amino acid, and now we have a completed protein. All right. So go to the drag and drop activity on my big campus or the website. Open it up and follow the directions and see if you can't go through the steps of protein synthesis, which includes transcription and translation, and build a protein similar to the way I just did. All right, I hope that helps, and I will see you in class, and I hope you have a great day.